So thank you very much for the organizers for inviting me to this talk. So as basically was just introduced, I will be presenting a primer on optimal transport. So the title is From Matchings to Optimal Transport Use Cases and Algorithms. But it will basically be a talk about why this theory of optimal transport has found relevance recently in various fields. Chiefly among them, of course, machine learning, but as we can see it more increasingly in biology, more specifically, for instance, single-cell genomics. So let me just make this as start with a big thank you to the community of people involved in this field. From the perspective of someone doing algorithms and, always being hopeful that the ideas that we are pushing end up being useful. I can say that this has been a big success. We've seen really very exciting applications of optimal transport to single-cell genomics in recent months, maybe in the last 2 years. And so a big thank you to the entire community and to all of you out there who are interested in those topics and are patient enough to read about the math that we're writing and being able to draw, the very interesting, exciting parallels and that make up those great new papers that I'm seeing recently. So with that, thank you. Let me just start with a very simple warm-up, just to set vocabulary right. So what is an optimal matching? What is exactly the idea of matching things optimally? So imagine you're in a space. So this could be a bit fairly abstract space, of course, but we could also be discussing very concrete matchings where you are red points and blue points. Here I'm plotting those red points as sticks and blue points as sticks because I would use later on the vocabulary of probability measures. But let's just assume you have, let's say, 4 red points and 4 blue points. They are in some space and you can somewhat quantify how close they are, how costly it would be to go from one to the other. The idea of coming up with a matching is essentially that of coming up with a permutation. So you have, let's say, numbered both the red points and the blue points, 1, 2, 3, 4, and you want to find a way to match one of the red points exactly to one and one only blue point. And you want to make sure that each of the blue points receives one match and each of the red points receives one match. So a typical example could be you have a few tasks that needs to be fulfilled. And you have as many workers that are waiting for your orders to start fulfilling those tasks. So let's say you have 4 tasks and 4 workers. And in that context, it makes sense to assign one task to one worker only and vice versa. And so to find a meaningful matching, you can start thinking about essentially, coming up with a permutation. So this would be the permutation that I just draw in the bottom here is just the one that I'm writing there. So you're associating to worker 1 task number 4, to worker 2 task number 2, to worker 3 task number 1, and to worker 4 task number 3. And because I have this prior information that tells me, for instance, that worker 3 is very close to worker 4, I am going to consider essentially as a cost function for that matching. This is an arbitrary choice, but let's say the sum of the lengths of those arcs here, so the sum of the distances. And obviously, if you look at this, for instance, from some aspects, this seems to be a fairly good match as far as Y3 and X4 are concerned, of course, because here X4, the worker X4 is assigned to a task, a Y3 that is very close to it. But for most other workers, this is not the case. They have to travel quite a long distance. So that's the entire thing about trying to seek an optimal matching and not just, let's say, a matching based on nearest neighbors and things like that. So you can start thinking, about different, of course, matchings, and there's going to be n factorial of them if n is the number of workers or tasks that are involved there. And so what I will refer to as optimal matching in the rest of this talk is this idea of, seeking for the permutation that minimizes the sum of distances between the worker and its assigned task. So that's just for the little warm-up. And now, why is this actually relevant in this talk? Well, we will see that there are various scenarios where both this matching this optimal matching will come very handy in some of the biological tasks that we will be targeting. And it's not only the optimal matching itself that will be handy, it's also a notion of distance, if you want, between those two. One could be tempted to call them sets of points. So you have, on the left, you have red points and blue points, let's say. So you're tempted to use a vocabulary of sets. So let's say a good notion of distance between red set and blue set would be this minimum cost over all possible permutations. But let me just explain a bit intuitively why it's not very useful to use sets. One of the issues with sets that you were aware of is that they kind of collapse, of course, when some points overlap. So in fact, what we will try to avoid and instead use is, we try to avoid this notion of sets and use directly, the notion of probability distributions. And so rather than looking at this as two sets, I'm going to just look at this as two probability distributions, because probability distributions are more accurate when it comes to, let's say, some of the points that are doubled or appear twice or three times, etc., or appear more frequently. So this is just a bit of a sometimes you will hear about people talking about optimal transport sets, but it's more like between probability measures. And let me also say something on top of this drawing, which is that there's essentially two different objects that, of course, are intimately related, which is the optimal permutation. So this is giving you the instructions on who to match with whom, and the notion of optimal transport or optimal matching cost. So it's just the difference between the min and the argmin. And I would like to highlight a bit that distinction now, because I will be in next slides using a bit that distinction. So on the one hand, this is the best cost you can achieve with any permutation, and sigma star is going to be the best permutation that helps you achieve it. So that's it for this small warm-up. And let me just now try to illustrate why this problem is relevant in biology, and why people have found it relevant in single-cell genomics in particular. So, of course, the audience here is far more qualified than I am in this field. And so this is really going to be a very, very uninformed sketch on my side. But suppose you have a biopsy and then you have a population of cells and you're culturing them. So either you're leaving them, on their own over time, and you want to watch them grow over time, or you are going to maybe apply some treatment and see, and you want to understand basically what's happening at the level of each of the cells. So, so I was saying that when you have these populations of cells, one of the goals of that you might want to is to reconstruct from this trajectories the individual trajectories of each cell. So maybe this is under some form of treatment, or just simply by looking at what happens with those cells. And of course, an extremely powerful tool to do that is given to us by single-cell genomics, which allows us to actually look at, for each cell, for instance, its transcriptome. And this would ideally allow us to be able to reconstruct those individual trajectories, of course. Ideally, we would like to be able, for instance, let's take one cell, measure it and put it back, etc. This is the way a machine learning person would think about this. Of course, the problem is that whenever we are going to make those measurements, we are killing the cell. So there's an obvious problem where we have an evolved population that is evolving with time, but whenever we actually want to measure it or have any idea of its features, we have to sacrifice that particular cell, and so we are not able to put it back in the population to track it individually over time. So this is where a very simple idea based on matchings can be useful. So you would, for instance, take a plate and sample a few cells from that plate at some point in time. So this would, let's say I'm taking 8 cells. This would give me, after processing them, a few points, in dimension, let's say 5,000. And with that in mind, I just remember basically which cells were processed and what feature characteristics they provided me. I would wait a few more hours. So in that time span, the cells would maybe grow, duplicate, die. So there's going to be a population who is going to vary in size. But I will, again, let's say, sample the same number of cells. So none of the cells that I had at time 0, of course, are represented in the plate at time 6 hours. But I'm hoping that, statistically speaking, the sample that I'm taking from that population is meaningful enough. And so that probably some of the cells that I took at time 0 had, let's say, siblings or related cells that were still present in the original population that were able to develop over time. And so this will give me another point cloud in the same fairly high dimensional space. And so the basic question that one can try to answer with optimal transport is if I had been able not to sacrifice the cell on the left-hand side at time 0, and I had let it live for 6 more hours, essentially, who might have become what, basically? What cell on the left what kind of cell at time 6 hours might have become any of the cells that I had measured at time 0? And so I think here we're going to a very crucial viewpoint in the application of the theory of optimal transport. What optimal transport theory is going to try to provide, and this is what is given in this idea of optimal matchings, is a rationale where you will try to cover all of the cells that you got at time 6 hours, the yellow ones, with the cells that you got at time 0. And in doing so, find an optimal matching. So we are really trying to aiming for a one-to-one correspondence. And somewhat in this very simplistic cartoon that I'm providing here, I'm expecting actually that each of the yellow cells on the right was caused somewhat, or not caused, but was driven from one of the cells that I got, one of the blue cells. So what this results in is a problem which I already presented to you a bit earlier, which is the optimal matching one. We will try to find a permutation that minimizes the sum of distances, and this will be basically my possible answer on what each of the blue cells could have become later when they became yellow cells. So this story sounds, I hope a bit convincing. What is a bit hidden here is the details of how you get the sigma star. And if you look at this mathematical formula below, what I'm essentially focusing is saying, okay, we'll look at for all possible permutations, associating one blue to one yellow. And the main input that I'm getting here is this notion of distance between cells. So in this cartoon, of course, it makes a lot of sense, right? It seems to be very meaningful, very natural to say that those cells are probably related if in this 3D space, they are not too far away. Because the devil is in the details, of course, this is going to be much, much more, let's say, far harder to carry out if you are in higher dimensions. So this is a bit of a caveat that I'm announcing. You should not see optimal transport as a universal tool to help you do this matching. It's going to be one that is essentially parameterized by the notion of distances between cells that you are defining. So in a way, if the notion of distance you define in the first place is rubbish, then the optimal transport is going to build on top of it and make something even worse. So as you will see in some of the applications of this to biology and other fields, it's always a bit crucial to be careful about how you set those distances there. So that's one first, I'd say, thing to keep in mind. But I hope I have convinced you that the story here is quite coherent, it's quite simple to understand. So this idea was essentially put forward in this very nice paper that is now about 3 years old. So I haven't gone through all of the details of what they do in the paper. It's a bit different. It would take a few more minutes to explain all of the nice modifications they did. But I guess the big picture is really this. Try to use an optimal matching tool to help you guess this who became what question. And let me just also do a bit of advertisement for a toolbox on which I've been working on recently. And Marius is going to mention the toolbox later in the second part of the talk. And so we've been trying to push a bit those notions of those ideas programmatically. And this experiment from the cell paper here is reproducing that toolbox. So the toolbox is called Optimal Transport Tools. And so we've tried to streamline that experiment and make it a bit compact in that notebook. So to open up a bit perspectives, I would like to start raising a few issues, with this very simple model that I just provided. And hopefully this will give an intuition on what I would say the computational community is working on to make sure those tools actually are useful. So the first problem that you will probably encounter is that this experimental description is very idealized because it requires that I have sampled exactly the same number of cells at time 0 and time 6 hours. And we all know that's very unlikely to be the case. So of course, there would be a very naive way to get around this, which would be to throw data. So if I have, let's say, 10 cells and 12 cells, then I would just throw randomly 2 of the cells that I got in time 6 hours. But that sounds not very clever. And so we need, of course, to provide a nice solution to this problem. And so you might imagine, basically settings where the number of cells is not going to be necessarily the same. And so you will need to do some kind of generalized matching. But if I actually say that, if I move away from this one-to-one matching, the hard one-to-one matching model that I've been talking to you about since the beginning, and I go towards something which is like, okay, I have a bunch of things and I just want to associate them and I no longer have this constraint of one-to-one matching, then you could think I could very well use something that is very, very simple, which would be nearest neighbor assignments. So for instance, any blue point would be assigned to a bunch of closer yellow points and any of the blue points and vice versa. So this is a bit, I'm trying to give you a sense of the spectrum at where optimal transport theory is going to lie. The problem with nearest neighbor assignments typically, and this is very well documented, is that we will have a lot of imbalance potentially. And so, for instance, you might be in a situation where a lot of the cells that appeared at 6 hours are essentially, if I'm just using their nearest neighbor, the nearest blue cell, then they would all be explained by just one single cell. That sounds very biologically plausible. Or vice versa, it would be in the situation where all of the cells that I had at time 0 collapsed into just one cell 6 hours later. And so you could argue that a lot of optimal transport theory and a lot of the works that have been that have been used recently in the field are essentially trying to strike a good balance between something which would be way too rigorous, I would say, which we one-to-one matching and not flexible at all, because you need to have the same number of points on both sides, and something that it's completely loose. It's just, let's just use nearest neighbor matches, and that's enough. You want to have something which is going to strike a good balance between not caring at all about the coherence of the story of the matchings and something that is really too hard. And so the idea here is the keyword that we use usually in transport is the idea of mass conservation. We are hoping that at least the amount of cells that are in some configuration in the yellow cell correspond loosely to a comparable amount of cells that were in the blue configuration. So there is other extensions that people can think about, which is the picture of the one-to-one matching is again very rigid, and also mathematically from a modeling perspective, it's also fairly poor in the sense that it doesn't provide you out of sample extensions. So of course, the first goal would be, can we try to connect whatever observations we had at 2 time points? But maybe down the line, what you would rather prefer to is to understand the entire, I would call this the vector field. Essentially, for a new point that would be arriving, a new cell that I just measured, what would be likely, state of that cell 6 hours later. So you can start seeing this evolution towards a more functional, I would say, out of sample approach. And then there is another very exciting, I think, development, and Marius will also be talking about this. The optimal matching setup assumed, as you remember, that I'm able to compare a worker to a task. So I could be able to quantify, for instance, how many hours a given worker might take to complete the task. So this means that I know how to compare things when I want to match them. There are settings where, it's notably in biology, where this is not the case. You might have some measurements taken from some cells in a given space, and measurements maybe 6 hours later taken with a different machine, for instance. And so they would provide you with two different representations, maybe not the same dimensions, for instance, or even completely incomparable representation. One might be discrete sequences, and the other might be, for instance, continuous vector. And so in that setting, you can ask yourself, is it still possible to match things? Is it still meaningful? And so there is actually a fairly intuitive workaround, and let me introduce it to you. So as we know, matching is essentially associating one blue to one yellow here. This is something we can do. I mean, we don't need a notion of distance to do that. You can propose an association. You can look at, for instance, the matched yellow points of two matched blue points. And as long as you're able to compare the distances of the points within their spaces themselves, which is usually easy. So as long as I can define a notion of distance 1 in the first space and distance 2 in the second space, you can essentially try to score how good a matching is, not by, again, comparing a blue point to a yellow point, but instead saying that if you're matching I to, let's say, sigma I and I prime to sigma I prime, you expect the distances to distance 1 between I and I prime to be not too far from distance 2 between sigma I and sigma I prime. So there comes the notion of this is called a quadratic optimal transport problem. And people have been calling this in the optimal transport community for quite a while, the Gromov-Wasserstein distance. And so you will likely encounter such definitions, and Marius will in particular talk about them next. So in all of the problems that I just covered, from my perspective, what I see is an intermediate matching or mapping step. So typically the matching is not, it can be, but it's not always the end goal. Maybe the end goal of this matching is just to disambiguate. The matching is just there to help you disambiguate things, and you want to do something next. And I really want to insist that, to me, this step is more like an intermediate step in the sense that more advanced learning procedures will build on top of it. And so if you want something that is able to do this matching at scale, to handle large dimensional data sets, I would say if you want to do this guided by a theory or some more higher order mathematics that help you inspire algorithms, and you want to make this in a differentiable way for end-to-end learning, then you essentially need to go towards optimal transport. So optimal transport is the theory, is like optimal matchings plus. It's really everything you wanted to know about optimal matchings and their generalizations. And because matchings happen everywhere, you will see that this field is basically present in various forms in various communities. And if I need to give back, some credit to the main people that started this, it is clearly from economics. So the story, the history of optimal transport really has its roots in economics, but it's also something that is now a very important tool in mathematics, both applied and pure. And it's also something that we're trying to sell a bit more increasingly to machine learning people. So we wrote with a book with Gabriel Perret recently, which is trying to cover some of the algorithmics of this field. So why is that? Why would you say it's a popular field? I think it's more like from the point of view of someone that is interested in algorithms, it really has a bit of everything. It is a field that will appeal to people that like, I don't know, partial differential equations, operations research, optimization, convexity. There's a lot of connections that arise in that field. And there are essentially, again, I'm repeating myself, but due to the fact that matchings are extremely important. And so the story of the field itself is, the field is attributed usually to Monge, the French mathematicians for the late 18th and early 19th century. And the reason I was insisting on economics earlier is that the field really became visible with the Nobel Prize in Economics of Koopmans and Kantorovich in the 70s. Algorithmically, this was solved for the first time by Danzig, who solved linear programs in general and the optimal transfer problem in particular. And then there was a big resurgence of a lot of mathematical works that started appearing in the late 80s, 90s, and 2000s, and which culminated around 2010 and 2018 with the Fields medals of Villani and Figali. So at this point, maybe, is there any question if I was not clear at some point?
Hi, I don't have a question, but one thing that occurs to me is that when you're looking at distance between multiple dimensions or across different data sets, you might have different levels of trust in those data sets or value certain dimensions over others. And I was wondering if you had explored using different weights for different dimensions or different data sets.
Yeah, so this goes back a bit to the warning that I put in the beginning. If you're not a bit careful with the distance that you are, the distances that you are providing, you will run into some issues. So it is a conservative stance, I would say, and this is the one that I see almost everyone take, to at least do a bit of data cleanup before putting those distances within the solver, the algorithmic solver. So I'd say this is a common pitfall, and people have to be a bit careful with that.
I have a question.
Yes.
How does this relate to like image matching problems in computer vision? Is it kind of the same thing, or?
No, I would say that. So matchings at its core, it's going to be about this optimal transfer problem. Then, when you're going to apply matchings in a given field, so in an image computer vision setup, this is going to be behind the engine of those matchings is going to be mostly what I'm going to talk about. So, for instance, there are, in image, you want to detect maybe objects and understand, for instance, what is in an image. And so typically there's been a lot of works recently that use a loss based on optimal matchings. So that you want to be, in a way, permutation free, basically. You want to remove, all of the ambiguity that you might have with a permutation. And so this is what is given to you by an optimal matching, engine running behind this. But then the applications usually are, they are all going to fall usually in this framework that I tried to present in the beginning. You have a bunch of stuff, and you want to associate them.
I have a question. What if your two data sets that you're trying to match lie on quite different manifolds?
You mean in the same space, but in different manifolds?
Yeah, or in different spaces. When you show on your slide, you have the intra distances, and you would with the quadratic problem. I was just wondering,
yes,
what happens if the manifolds are quite different?
Yes, so typically then in that case, it means that the distance between them will be high. You will have a hard time finding this sigma that will make this distance small. So this is a very good point. Essentially what this, so here I just put the sigma star, the argument. If you look at the minimum value that you will reach, this is essentially quantifying how hard it is, how not isometric those two spaces are. So if they are different, indeed, you're going to have a hard time matching them. So if I basically how hard it is to match those manifolds despite the fact that they're in different spaces. Yeah. So
hi Marco.
Yes.
Question from the broad.
Yes.
So also just to say first, this is a very beautiful primer, really clear. Really, everybody here is enjoying it. We have a good crowd here in person. So quick question, you highlighted this kind of spectrum from the very rigid of one-to-one matching to the super uncontrolled where we can get kind of anomalies of things being matched to too many things and so on.
Yes.
And I was wondering in this kind of in-between, is there room for imposing some kind of prior knowledge rather than saying we kind of expect things to, kind of have the same mass or whatever? Is there a way to say something a bit more specific? How hard is that? If we have some prior knowledge, how hard is it to incorporate it?
So how would you quantify? I mean, I'm sorry to answer with a question, but the prior knowledge that you would have, what form could it take?
Maybe a form like we expect certain populations of cells to maybe disappear from one point to the next,
yes,
or some to expand dramatically. So like we have some idea of a non-equal kind of distribution of neighbors, but maybe for some we don't know. So maybe it's more the sense that we have an unequal amount of uncertainty about the number of neighbors we want to match.
Sure.
Across different classes.
I can also imagine some kind of, some areas of the space would be more prone to growing and some areas of the space would be less prone, something like that maybe also.
Oh, sorry, I missed that.
Yeah, so I was saying some areas of the space would be more prone to growing mass and some areas of the space would be less prone, let's say.
And at the same time, for other areas, we would want to stay unbiased and know
yes.
nothing. Yep.
Yeah. No, this is a very good point. I think to start encoding this, you probably need to look at the different ways to penalize for deviation, of course. So this is going to be a standard machine learning probably answer. If you have prior knowledge, then you usually would turn into some form of penalization.
Makes sense. Or maybe like a weighting in those like optimal matchings within space or something like that.
Yes, or weighting the cost functions as well, right? So
yeah, exactly.
I would say that the model is fairly simple. So it doesn't have that many moving parts. So you can play with either the cost or regularizations on the couplings.
Right. So the standard machine learning like way of that makes sense.
I would that's where in the absence of other elements, I would probably go for that.
Great, thank you.
You're welcome. So let me just go back to this picture. So the generic, so the picture I showed you earlier, with the 4 red points and the 4 blue points, as I said, it has a few limitations. So when the number of points is not the same, and then we just had this comment, what happens when, for instance, the mass of some of the points is not the same? So we cannot always necessarily start from the principle that all of the points are equally weighted. So the standard optimal transport problem, the more flexible formulation of this matching problem, is to assume that we have xi's, the red points, and we have ai's, and we have bj's and yj's. Okay, so this is the second measure, the blue measure, and we will try to find a way to transport this optimally. And so if you encode things as a transport and not so much as a permutation, the natural way to address this, or this could be seen as a relaxation of permutations, is to encode things in matrices. So on the one hand, you will have a matrix that will be containing all of the distances between the red points and the blue points. So I'm adding this power p there because often people like to put this power p, but please don't be distracted too much by it. P can be 1 or 2 or 3. It's an integer that's bigger than 1. And you have, on the one hand, this cost matrix, which is going to store all of the distances, pairwise distances between the points, so xi's and yj's. And on the other hand, we're going to consider a set of matrices So we're not going to think in terms of permutations anymore, n factorial permutations. We're going to think in terms of matrices, which are of size n times m that have non-negative elements. And for now, I'm going to assume that for a matrix P to make sense, it needs to have row sum A and column sum B. So what does this mean? It means that you have a matrix which describes at each entry ij how much of the mass of the red xi point has been assigned to a yj point. And so if you take this view, then it means that the row sums for the mass conservation to work, you can't send more or less than what you have at each of the i's, and you can't receive more or less than what you need at each of the j's. So you need to have a row sum which is equal to A and column sum which is equal to B. And the optimal transport problem next is just simply this simple linear this dot product between P and the matrix MXY. So it's going to be the sum of all the Pij's, distances between Xi and Yj's to the power P. So if you take a step back, this is really what people in optimization call a canonical linear program. And what we know is that from linear programming theory, we can derive from this this linear program another linear program called the dual optimal transport problem. And the dual optimal transport problem results in the same quantity eventually, but it's parameterized differently. It is going to be, instead of having a rectangular matrix that provides you how much mass is transferred from each point yi to each point j, it is going to be defined in terms of two vectors of prices of size n and m respectively. So one vector for the size of the first set and m for the size of the second set. And we are just going to maximize the dot product between alpha, the prices, and A, and beta and B. So why do I call this prices? Well, it's essentially because this is going to describe the optimal transport in a different way, rather than focusing on the procedure of sending things from ij. We're going to focus on the pricing of sending those goods from the first measure to the second measure. And so the total price is going to be a price per unit of good in the first measure. Is going to be alpha I times the amount of goods AI and beta j pj. And there is a natural constraint that arises there, which is that the price that you can come up with to pick some good at I and deliver it at j, so alpha I plus beta j, cannot be bigger than the distance itself. So this gives you two different perspectives on optimal transport problem. Again, let me just conclude with this. On the left, we have this, what is sent to where? And on the right, we have, okay, after resolving this transport problem, I have a set of prices which tells me it is sending that the mass from each of the blue points on the lower left has this price, and receiving it on the red points on the upper right has this price. If you're familiar a bit with duality theory, what is very nice about it is that it provides you a handy way of understanding essentially how the problem and its optimal values are going to change when I am going to modify the inputs. And some of the inputs here that can be modified are either the locations of the points on the left, or this will be the useful part on the right hand side, when I'm going to modify the amount of mass. So this is just a parenthesis on duality. So if I just look at this problem again, this is a linear program. A linear program is always going to be described in the slide as, some direction, a set of stuff, that is valid, and you need to pick one point in that set that has the lowest dot product with that direction. And so here in this case, I think it's very clear. If I'm drawing this yellow blob, all of the matrices that have the right row sums and column sum constraints, and on the other hand, I have this cost matrix on the top left that is providing me the direction, the P star is going to be this vertex, the vertex of this corner. As a side note, this is cubic in the number of points. So it's very costly. It's a half full or half empty glass. If you're coming from optimization, it's actually a fairly cheap price tag. But if you're coming from applications, it's way too costly. What I have been working on in recent years, and I'd like to also focus on, is that this is not only problematic to have such a big price, it's also problematic because the solution P star is not very stable in the inputs. And let me just try to show you this in a cartoonist way. Imagine, for instance, I'm changing a little bit, my x's or my y's here for a while. And so as you've just seen, for a few seconds, nothing happened. The optimal solution was clearly the same vertex. But then all of a sudden, I hit, my direction is orthogonal to a facet of this set of matrices. And then I jump to another solution. So in practice, what this means is that the distance is not differentiable and the solution P star is unstable. It's not always unique. And what is going to be the most problematic part is not so much that you're going to be orthogonal to a facet, because this happens never. It happens with probability 0. The problem is you're going to jump from one solution to the other. And so if you want to use a bit more mathematical terminology, this means that the way this P star matrix moves or changes with the data is extremely is badly behaved, because as you've seen, either P star did not change at all for a while when I was changing my x's, or all of a sudden it jumped to a completely different value. So there's no meaningful Jacobian with respect to the inputs. And let me just pile up a few more issues in terms of algorithmic resolution. To solve this problem, you need to use this family of network flow solvers, which are essentially very complicated, fairly complicated, beautiful, but complicated combinatorial solvers. So by saying this, I'm hoping I'm giving you a picture of, the picture should be as follows. Optimal matching is cool. It's nice. It's important. It can help you disambiguate, whenever you don't know who might have become what, as long as you have a good notion of cost. But first, it's going to be very hard to compute in practice because it is very costly in terms of computations. It's cubic. And let me also point out another source of issue, which is more statistical. So in practice, most of us, even in the single-cell genomics example that I provided, we will always think about data as sampled from a certain density. So you would say there's a probability distribution of cells at time z, 0 hours, and I'm sampling, let's say, 1,000 points out of that. And so we always have, this model where we're sampling points from a idealized density. And so the question is, if you were to be able to compute, the optimal transport distance or the matching between those two samples, fine, that's already an achievement. Maybe it's going to be costly, as we just mentioned. But if you go beyond that and think of it in statistical terms, what if you had access to the entire densities or the entire probability distributions? And let's assume this transport problem that I just described extends very naturally to the continuous setting. How different would, on average, be the result that you would want to compute for the entire density from that you have through the samples? And in other words, this is called the statistical complexity of the problem. Is it really worth it to sweat, and work hard to compute this linear program if, on average, the result of that linear program computed on samples is very far from what you get on the true densities? And unfortunately, the news is bad on both sides. So it's not only actually very hard to compute this, it's also well known and very well documented that if you just follow, the Kantorovich, the Nobel Prize in economics route to compute optimal transport between two densities, and you're only looking at them from their samples, you're essentially going to work very hard to overfit whatever samples you have. So in other words, in data sciences, and this includes really single-cell genomics, we must really regularize whatever matching or optimal transport problems we have to improve on either or both aspects. And those are computational aspects and statistical aspects. So this is really a crucial, I think, message of this talk. If you are aiming at using optimal transport in your biology pipelines, it's great to look at the original math books for inspiration, but please keep in mind that we are not in their regime. Our regime is really that of using data sets, finite samples, and so we have to regularize those problems. We can't just directly import whatever optimization problem was defined by Kantorovich in the 40s. We need to do something a bit different.
Marco, quick question. It looks like that last problem also makes sense for the argmin. So it's not just the value of the Wasserstein distance, but does the map extend? It's part of I guess it's really just a continuous extension question you mentioned, if the densities themselves are continuous and
yes. So
it seems quite like an interesting problem. It sounds like you're saying that the general answer is not so good unless you do some regularization.
Yeah, I thank you very much for pointing that out and for pointing out the distinction. So there has been some work by Philippe Rigolet, who's at MIT recently. So he's more interested in min-max results. So this is going to be like a min-max optimal estimator of this matching. Let's say the mathematical word for this is the Monge map. And so again, this is a bit hopeless, and this is also going to be impacted by this curse of dimensionality as well.
Yeah.
So it's even worse. I would say the argument is obviously going to be even more unstable. And so definitely we're trying to also, of course, this is a very good point, try to get around that issue as well. So regularizing is even more needed, I think. I would even say if you are going to build on that sigma star, regularization is even more important, I would say.
If you want to try to learn what sort of continuous mapping between the spaces extends the optimal permutations as the data sets grow. It's
even more critical, I would say.
Yeah, no, that's intuitively that makes sense. Thank you.
Yeah, thank you. But thank you very much for pointing that out. Thanks. So I'm running a bit short of time, so I'm putting a lot of burden on Marius to introduce stuff afterwards. But let me just very briefly introduce this idea of regularizing the optimal transport problem. And there's been a lot of work on this in the last, let's say, 7, 8, 9 years. So this is the reason why I think the field is still very excited, because we do what we as machine learners did what we know how to do, which is regularize whatever, take a problem and regularize it in as many ways as we can. And there's one route that is actually very intuitive and very simple to do that regularization, that is entropic regularization. So entropic regularization is just simply the idea that when we're computing those optimal transport matrices, which are those relaxed permutations, we are essentially computing a probability coupling. It's going to be a matrix that has row sums equal to a probability distribution, column sums equal to a probability distribution. And so it's very natural to regularize the probability distribution by its entropy. So here in this case, what we'll be adding is minus the entropy, so the neg entropy. And what this makes is that this turns the problem into something completely different, from a linear program to something that is essentially a convex problem. And it's a gamma strongly convex problem we can prove. And so intuitively what we get by adding this term is imagine you are computing optimal transport between those two 1D distributions and then plotting here in the middle this matrix, which is the transportation plan. It's actually a bit hard to look at, the entries. They're very thin, so they more or less look like this, let's say. And what you're getting in exchange for this gamma entropy, neg entropy regularization is a bit more blur. Remember, if you want, of course, higher entropy, which is putting its minus in the min, it means you want more blur in a probability coupling. But it has to be a blur that is applied in relevant areas. It's not just blurring with an isotropic Gaussian blur that would kill the marginal, the constraints of the masses. And so what you were aiming for is a good trade-off for areas where we can trade off a bit without losing too much in terms of the cost. And so you would see this kind of behavior. So let me just say that the reason people like this regularization is that it is very efficient from a computational perspective. And it has been powering a lot of the approaches that we've seen in optimal transport recently, and notably this Cell paper that I cited earlier on. And so let me just be a bit brief with the math. This is a convex problem. So this is fairly standard. You can use something called the Lagrange method of multipliers to include the constraints of the row sums and the column sums. And if you just work out the math, you find out that the solution needs to have a factorization, which is explicit, except that you don't know the vectors u and v that help you factorize this matrix. But it's basically going to be a diagonal on the left times the matrix k, which is the exponential of minus the cost matrix that you had. So this is the element-wise exponential, and diagonal of v. How you get there? Well, just trust me. We'll share the slides. It's just first order methods. So the first order differentiation, Lagrange method of multipliers. And so we get that you have this factorization. And once you have this explicit factorization, the game is trying to find the vectors u's and v's that help you satisfy the marginal constraint. So this means that I want to have a u and a v such that this matrix, diag u, k, diag v, has the right row sums and column sums. So if I just turn this into equation on the right, on the lower right, it essentially means that I want to satisfy two sets of nonlinear equations. And if I do a bit of cleanup, diagonal matrix times 1 is the vector, and then diagonal matrix times a vector is a vector times another element-wise. So I do a bit of cleanup. It means I need to find two vectors u and v that satisfy these two. I'm saying nonlinear because v here appears as a denominator on the right, and or u on the bottom. And so this is essentially what we need to have. And there's a very naive but surprisingly here powerful way to do this, which is to repeat an iteration which looks like this. You just start with a random v, and then you apply those two iterations. And the reason we call this a synchron algorithm is that this was proved to converge in the 60s by Synchron, a mathematician. So to give credit to actually other people, this predates Synchron by a lot. This was found and discovered in the early 20th century by economists again, and was known as the RAS method. It's also called the IPFP method. So it's been rediscovered in many contexts. But what is quite exciting about it is that we are actually improving our understanding recently about this algorithm, because it has become really a very important workhorse for optimal transport. So if you want to use those matchings, it's likely you will be using this form of algorithm. And there's a lot of nice things about it, which is that compared to the very highly complex, combinatorial algorithms that I described earlier for network simplex, this is extremely simple and parallelized very well on GPUs, because the main computation effort needed here is just a matrix times a vector product. So this is what I wanted to say about the Synchron algorithm. Let me maybe move a bit faster, because I know that Marius needs a few things about it. I have a few slides on differentiating the Synchron algorithm. Those are the slides I won't be able to present, and also about some of the properties of how this was going back to the question how this P star will change when I'm going to change the input values. But I also wanted to introduce two nice generalizations that people have been playing with, specifically within the field of single-cell genomics. And the first one is the idea of getting away from this mass conservation. And this goes back to the question that was asked earlier. And so the first attempt at doing this was considered about 5 years ago, is to say, well, the optimal transport problem is this minimization on P with row sum A and column sum B of this linear product. What about, again, regularizing for this constraint rather than having it as a hard constraint? Why not just penalize things? And so people discovered this approach and called this the imbalanced optimal transport problem. And essentially they would say, let's just consider matrices that are size n m, non-negative, but instead of having a hard constraint that they have row sum A and column sum B, I am going to penalize with a row term The Kullback-Leibler divergence, you could use, of course, many other ways of penalizing, but Kullback-Leibler is actually quite natural. The Kullback-Leibler divergence between the row sum of P and the column sum of P. And what is very, very nice is that if you add, again, this magic entropic regularization on top of this unbalanced formulation, it is very trivial. You recover exactly the same synchron algorithm that I showed you earlier, except that now at each iteration, you will make a very small modification. And the modification that is that you will raise to a certain fractional power the iterates that you get. And let me just, maybe to convince you a bit, if you let rho go to infinity, meaning you fall back on the hard penalization for the row sums and the column sums, then you will see that this fraction here goes to 1. So everything kind of makes sense. Another regularization, or let's say another formulation that we've come up with recently with co-authors, is to use some form of explicit factorization of the solution P. And so rather than actually having a solution matrix P of size nm, we have recently proposed to factorize P as a product of non-negative matrices. And so this is still still relatively new. So I won't have too many resources here to present it, but as Marius will mention later on, this is very promising because this explicit factorization, much like, let's say, non-negative matrix factorization, allows you to play with the notion of rank. And so instead of having to do computations with nm values, you can only do them in linearly with respect to number of points. So to sum up, optimal matchings are used everywhere in machine learning. And they're used both to compute distances, but also to do this registration, which this is the sigma star that I was alluding to. To make this work, really, we need regularization, because this is the only way to both make sense statistically and also make sense to scale up, computations. So I've proposed a few regularizations. The first was this entropic regularization, then maybe regularizing for marginal deviations, and then the other approach is this different set of constraints for low ranks. And I haven't had the time to talk too much about this, but this is personally one of my focuses of interest is differentiating this, so being able to predict how matchings will change when the inputs changes. So thank you very much. I'm aware I'm maybe a few minutes over schedule. I'm sorry, Marius.
Thank you, Marco, for the really nice primer, also from my side. So this talk is going to be about applications of optimal transport, in particular about a toolbox called Moscot, which is a toolbox for scalable optimal transport applications in single-cell genomics. So here's a quick overview. Anna basically said it already. I studied physics in Freiburg in the southwest corner of Germany, as well as in London. For my master's degree, I switched to applied mathematics. Doesn't seem to advance, but I can just go here, I guess. Okay. Okay, and now since about 4 years, I'm doing the PhD in Munich in the lab of Fabian Theis. So I'm coming to the end and I'm currently writing up. Also went on 2 lab exchanges actually in the PhD. The first one was not too far from here in New York to Dana's lab. And the second one was just last year where I visited the lab of Monit Zahn and where this Moscot project also started. So this talk has 4 parts. The first one is an overview of optimal transport applications in single-cell genomics and current challenges as I see them. The second part is about our proposed solution, the Moscot framework. And then in parts 3 and 4, I go into applications of Moscot to mapping cells across time and space. So let's start with this first part. Optimal transport has found quite a few applications in single-cell genomics. Marco mentioned this top paper here from Jeff Schiebinger, the Waddington OT method, which has been extended in many different ways since the original method. For example, here in lineage OT and also in super OT towards using barcode information as well, and also towards different modalities here in that method. There have also been applications to spatial problems, to data integration. We also saw this Kummer-Wasserstein application here before, to learning patient manifolds. So basically using optimal transport to describe a patient level distance in terms of underlying single cell samples and to mapping cells across different perturbations. So this basically shows that optimal transport has been proven to be a powerful and versatile tool for many applications in single-cell genomics. Next, I want to go into the different flavors of optimal transport a little bit. Marco talked about the classic optimal transport setting where you have two densities that live in the same metric space where you can do one-to-one comparisons. But Marco also mentioned that there are often situations, also in single-cell genomics and in other data science applications, where you cannot make one-to-one comparisons between the two different spaces, for example, because they have different dimensionalities, or in general, because they're just two different metric spaces. And for that, you can use Gromov-Wasserstein optimal transport. And I want to illustrate this in another example. This is an example from mapping cells from a reference single-cell RNA-seq sample, as you can see here. So we have N cells measured in G genes to a spatial target domain. So the original paper that explored this idea with optimal transport here is the one from Monet-san, Nature 2019. And in this case, actually, we have no spatial information. So let's just assume we just have the target domain. We know what the geometry is, but we haven't measured any markers in the spatial domain. Still, we want to find a mapping. So we want to find this coupling, usually this entropically regularized coupling between the two spaces that basically assigns each cell a distribution over spatial positions. So in classic optimal transport, we would now have to define a cost. Marco called it M before, I now call it C, between cell I and location J. But of course, in this context, it doesn't make sense. What is the distance between a cell in transcriptome space and a spot in space where you have no gene expression information? That doesn't make sense. So instead, you can use a trick. You can say, I can define a distance in transcriptome space between pairs of cells. And I can also define a distance in my spatial domain between spots or whatever my resolution is. For example, just use Euclidean distance in that space. And then I can make a structural correspondence assumption. I can say that on average, for most genes, for most cells, two cells which are similar in gene expression space should be mapped to two spots that are similar in, say, Euclidean distance. And in the original paper, Moore and colleagues validated that in the examples they looked at, for example, here in the Drosophila embryo, this assumption actually makes sense. So on average, as you increase spatial distance, expression distance also goes up. Now, this can be put into the Gromov-Wasserstein loss function that Marco also showed before, where you have the two cost matrices here between your reference in gene expression space and your spatial target domain. You have a loss function, which before was Euclidean, but you can also explore different ones. And you have your entropic regularization again. So this would be your Gromov-Wasserstein, the quadratic optimal transport term now, and that is the regularization. We can go one step further. We can take this idea of Gromov-Wasserstein optimal transport, and we can combine it with the linear optimal transport. This is called fused Gromov-Wasserstein, and in this example, it would lead to better spatial reconstruction. So now let's say that we don't just have access to the spatial target domain, to the geometry, but we have actually also measured a few marker genes, in this case, only 4 marker genes. So we can do the same trick as before. We can define a distance here. We can define a spatial distance in that domain, but now also within those genes that are shared, so in this case, gene 1, 4, 17, and gene G, we can actually make one-to-one comparisons between cells in transcriptome space and, well, between cells in the single-cell RNA-seq sample and between cells in the spatial sample. We can still make the structural correspondence assumption, and now we can combine the two pieces weighted by an alpha factor that is now a weighting factor between the Gromov-Wasserstein, so the quadratic term that enforces the structural correspondence assumption, and the Wasserstein, the linear term that we saw before with Marco. And again, we can add entropic regularization here. So that flavor is called fused Gromov-Wasserstein, and it will be important in many of the applications that I show later on. And this idea is not new, it's not for me. That has been first shown to work very well here in this setting in the NovoSpark algorithm. Okay, now we can look into how the different methods fall in these categories of the different flavors of optimal transport. So most use the classic, potentially entropically regularized, potentially unbalanced version of optimal transport, such as Waddington OT. But there have also been Gromov-Wasserstein applications, fused Gromov-Wasserstein applications, and some more exotic flavors of optimal transport, such as Berry centers, neural OT, surrogate OT, or convolutional OT, which are not really the focus of this talk. So in this talk, I will focus on classic OT, Gromov-Wasserstein, and diffuse Gromov-Wasserstein problems. So OT in single-cell genomics is so versatile because it has different flavors that can be applied to the different situations that come up in problems. These different flavors are usually linked to different implementations. A very popular backend is the Python optimal transport toolbox that most of these methods use, but there are also custom implementations in NumPy or PyKops, where KOps is sort of a very fast C library to do kernel matrix vector multiplications. There are PyTorch implementations in R. So the implementations of the different sort of optimal transport flavors and also of the different biological problems that build on top of optimal transport are split across many different backends, which leads me to the current challenges of a truly community-wide adaptation of optimal transport in single-cell genomics. The first one, from my point of view, is a sort of fractured tools landscape. So while optimal transport is very versatile, many of these implementations actually don't interface, for example, with n-data objects. And once you are familiar with one of these implementations, if you want to solve a different biological problem that under the hood uses the same machinery of optimal transport, you have to get used to a different API. Secondly, scalability issues. As Marco mentioned before, even though if we regularize the optimal transport problem, we go from a cubic complexity to a quadratic complexity, that is still not great in practical applications. And for Gromov-Wasserstein and fused Gromov-Wasserstein, it looks even worse because there the complexity is cubic, and it's cubic already when we have regularized. So it doesn't get better than that easily. So there's scalability issues in practical applications, and there's limited support for multimodal data. So most of these methods have just been formulated. Of course, there are exceptions, but most of them have just been formulated for simple one modality data like just RNA. For this, we propose MOSCOT, which stands for Multi-omic Single-Cell Optimal Transport Tools. MOSCOT comes with a unified modular API, which for us means from a user perspective, it gives you streamlined, but mostly also very convenient and easy access to OT-based solutions. And from a developer perspective, it's easy to extend in two directions, either towards new models. So say you want to define a new biological problem that you can solve with optimal transport under the hood. If you implement that in MOSCOT, you can automatically make use of all the machinery that we have in the back end, like the fast solvers, or also the APIs to n-data objects and so on. But if you are a developer and you have a new optimal transport method that is maybe faster than previous approaches, you can also implement that in MOSCOT, and then it's automatically available to a subset of the biological problems that we have defined. It's also scalable in both time and memory, as I will demonstrate in some applications and some benchmarks later on. And it's applicable to multimodal data. And we're trying to build a mechanism to have consistent support for multimodal data across many of the different models. So these two things here, the unified modular API and the scalability in both time and memory, that's something I want to demonstrate and focus on in this talk. And the applicability to multimodal data, that's currently work in progress and not so much the focus of this talk. So that brings me to the second part already, the MOSCOT framework. But before I actually want to introduce the MOSCOT team that has been working on this, from Fabian's lab, there's Giovanni, there's Mike, there's Dominic. Then we are collaborating with Bastian from Jan-Philipp Junker's lab in Berlin. And from Fabian's lab, there's also Manuel. Then from my lab exchange at Moritz Science Lab, we have Zoe on the MOSCOT team. Of course, there's Fabian. And then we're collaborating on the OTT side, which is in the back end, as I'll just show in a minute. We're working together with Marco and with Letizia. So let me show in this slide what MOSCOT looks like. So basically, each box here corresponds to a collection of classes that either the user interacts with or developers interact with. So as a typical user, you will interact with these domain-specific problems that we have defined in MOSCOT. These are then translated to an OT problem by the optimal transport problems class. So I think you can't see this very well here. So essentially, these are translated to optimal transport problems by this OT class, and then via a solver interface, they are sent to an OT backend that solves these problems. Then the solution is passed via a solver output interface back into the domain-specific problems, where you can analyze them using analysis mix-ins. Currently, we're focusing on temporal and spatial problems in Moscot. That's also what I will demonstrate in the applications later on. To define these problems, so to set up these problems and also to provide downstream analysis functions, we interface with the wider ScanPy ecosystem, so ScanPy, SquidPy, SCDI tools, ANData, and CellRank. And the problems that we currently support in Moscot are classic OT, Gromov-Wasserstein, and fused Gromov-Wasserstein, and we're currently extending it towards Berry centers as well. In the backend, we are based on OTT, so that's the framework that Marco highlighted before, that he built together with his team. I'll say a few words about this also in a minute. In the future, we plan to extend towards Python Optimal Transport Toolbox and geom-loss, which is in PyKOps as well. And I want to highlight Dominic and Mike here, who've been working on this implementation a lot. So what does it mean to have optimal transport tools in the backend? Well, it's JAX-based, which means you have GPU acceleration, just-in-time compilation, and differentiability throughout the entire OTT toolbox. And it also means available in every of the Moscot models. So for example, for every biological problem that we define in Moscot, you can always solve it in a GPU. It doesn't matter whether it's a spatial problem or a temporal problem, you have consistent GPU support. And also, one thing that comes on top are these low-rank factorizations that Marco briefly mentioned at the end. And I will also show what that means in practice in some benchmarks. It's not yet public, but we're currently working on the GitHub release. So that brings me to the first set of applications, which is mapping cells across time. That's this classic idea that, that Jeffrey was working on in Waddington OT and that Marco also used as an example. So we have two time points, for example, time point 0 and time point 1, and we want to map cells from the earlier to the later time point. In Waddington OT, that is formulated as an unbalanced and tropically regularized optimal transport problem. And once you have solved that using optimal transport, you can ask biological questions like, what is the likely descendant distribution of these cells here? What are likely ancestors of these cells? Or questions about shared ancestry. When do trajectories diverge? When are fate decisions established? In Moscot, it's very easy to do that. I will demonstrate these code snippets throughout the talk. They always look very similar. So basically, you import this problem from the Moscot interface. You set it up with an ANData object. You prepare it by passing some information that you need to solve this problem. And then you solve it by passing, for example, the epsilon value for the entropic regularization. And once you've done that, you can make use of these downstream analysis mix-ins to, for example, look at ancestor or descendant distributions. All right. So in practical applications, scalability matters because the size of these data sets is ever increasing. And as Marco highlighted before, the time complexity of classic optimal transport is quadratic, but not only is the time complexity quadratic, also the memory complexity is actually quadratic. And that's because this K matrix here, the Synchron-Gibbs kernel that we saw in the Synchron algorithm before, is defined as minus the cost matrix C over epsilon. And C has dimensions n times m, where n is the number of cells in the source distribution and m is the number of cells in the target distribution. So the bottleneck in Synchron is these kernel matrix vector multiplications for these two scaling vectors here. Okay, which means that if you pre-compute the kernel matrix, what you essentially do is say we have just 2 cells in time point t0 and we have 3 cells at the later time point. We have to make all versus all comparisons and then store all of them to compute these matrix vector products later on. And this gives rise to a space complexity of nm, which becomes prohibitively relatively soon, especially if you think about GPUs, because we can natively run this on the GPU. If you have large cell numbers, your GPU will pretty soon be out of memory and you simply cannot run it for large cell numbers, even though it wouldn't be a problem from the temporal perspective. The time complexity would probably it's relatively fast to do this on GPU. You just hit out of memory pretty soon. However, if you think about it, actually, if we're just interested in computing one element in this output vector here at a time, say the first one, all we need to have access to is one corresponding row in this kernel matrix here that we have to multiply with the vector V. which in this picture means just fixing one cell on one side and making comparisons with all cells at the other side. And that, of course, has linear memory complexity, because I only have to hold these samples here in memory. So if you do that, which is something that's called online memory evaluation in OTT and is available through OTT and also in Moscot, then the space complexity is reduced to m plus n times d, where d would be the dimensionality of the data. And typically, we define these cost functions in latent spaces like autoencoder latent spaces. So d is between 10 and 50, so it's not too high dimensional, actually. So I want to emphasize that this online evaluation is not an approximation, it's exact. The result that you get is mathematically equivalent, it's exactly the same. It just resolves this memory complexity, which really enables you to run this for much larger data sets. So here's a benchmark. We started actually by looking at this memory complexity for now on CPU, because it's a little bit easier to profile memory on CPU. And we compared Moscot, the online version of Moscot, which corresponds to the online version of OTT in the backend, with Waddington OT, which has a little star here because I'll show some GPU benchmarks later on as well. But since Waddington OT can't be run with the GPU, we'll always run it on CPU. So if you compare memory here, you can really see that, the online implementation has a much better complexity. For the final cell number, which was, how many cells was it? It was 164,000 cells. So for this number of cells, Waddington OT takes around 230 gigabytes of memory, where Moscot in the online version takes less than 2. So if you think about GPU memory, this is something you can easily run on pretty much any GPU. Whereas if we wouldn't use this online version, we would need 130 gigabytes of memory, which becomes prohibitively expensive very soon. We then checked time on the CPU. All the methods are relatively similar there. Online evaluation is a little bit slower on the CPU. On the GPU, though, we saw that if you just run Moscot on the GPU without online memory evaluation, you hit out of memory relatively soon because it has this quadratic memory complexity. If you use the online method, though, you can really go to large cell numbers. So in this case, Waddington OT took 52 minutes. This is a little bit larger than before because now it was a different CPU. So we run all methods on the same CPU in that benchmark here on the left. On the right, since we used our GPU clusters now, it's a different CPU that was used for Waddington OT. So it was a little bit slower, took 52 minutes here. Moscot couldn't be run in the non-online mode because we hit out of memory, whereas Moscot online computed the coupling in less than 1 minute for 164,000 cells. So it's really extremely fast, and it really scales to entirely different dataset sizes compared to what we had before. Now, it's really fast, but we still have the quadratic memory complexity. So if you would go to much larger dataset sizes, at some point, this quadratic term would kick in and time would basically explode. So with the online evaluation, we resolve the memory bottleneck, but we don't really resolve the time bottleneck. We've just sort of delayed the point where this quadratic complexity kicks in. If you want to solve the time bottleneck as well, we can use the low rank approaches. Marco briefly introduced them. Basically, they're equivalent to saying, not only do I want my coupling to come from this space of admissible couplings here defined by the marginals, but I also want them to have at most a certain rank here. So we use this explicit factorization, or OTT uses this explicit factorization, and we say the rank of the feasible set of admissible couplings can be at most R. And if you do that, actually, you get both linear time and linear space complexity. So there are no more n times m terms in either the space or the time complexity. They are all order n plus m now. We looked at this in a practical benchmark. We compared the same 3 methods as before here on the left. Now we also added a few low rank approaches. In terms of memory, the low rank approaches all scale extremely well, about as good as the online evaluation. Depends a little bit on the rank, what the actual memory requirement is, but they're all extremely low. And in terms of time, they're also extremely fast. You can see that here towards the end, the time requirement for the online method goes up because the quadratic complexity kicks in, whereas low rank is still extremely fast and can solve the entire problem on the 164,000 cells in less than 1 minute. Now, of course, while online memory evaluation is mathematically equivalent to what we had before. Using low rank approaches actually is theoretically a little bit different. And even though there's very nice theory behind this, I highlight 2 papers here at the bottom. We wanted to make sure that in a biological example, it gives sort of similar accuracy. So we looked into the original Waddington OT paper where Jeffrey and colleagues did a comparison. So they did something they call geodesic interpolation, which is you take 3 time points in a time series experiment, you leave the middle one out, and then you try to approximate the middle one by computing a coupling between the outer 2 time points. That's what they call geodesic interpolation. And then they compare basically the distance they get from the interpolated time point to the ground truth time point that they held out. They compare this, or they computed this for their optimal transport implementation, and they compare to two null models that essentially correspond to random guessing, the coupling, and to one baseline, which is the batch-to-batch variation within one time point, which is sort of a soft lower bound on how well you can expect to do on this problem. I focused on these last time points here, where the difference was pretty pronounced, and compared different implementations. So Moscot full rank here, there would be the method that is equivalent to Waddington OT in terms of accuracy. And you can see that when you use sort of ranks between 10 and 1000, you get similar accuracy. When you go to really low ranks, like rank equals 1, you get an accuracy that's about as good as random guessing. So this basically confirms that A, it's relatively robust to the exact rank that you use. And as long as this rank that you use is high enough, you get an accuracy that is comparable to what you would get if you would run the full rank optimal transport problem. Okay, still there are some limitations to this approach. One is that it works very well when the time gaps are not too far apart. Already in the original paper, though, they showed that when you increase this, so when you artificially remove some of the time points, when you go to 2-day time gaps, then it doesn't work as well any longer. So the geodesic interpolation distance becomes much bigger. And also in another paper from Malone-Klein, they showed that if you have hidden variables, for example, on the epigenetic level, so if fate priming is established on the epigenetic level rather than on the RNA level, and you don't measure the epigenetic level, then if you use an approach like Waddington OT, these couplings will not be very informative in the sense that they don't agree very well with ground truth given by clonal labeling in this data set. So the correlation is really low. So in settings like this, you need additional priors to get biologically meaningful couplings between time points. And one prior that we were exploring is actually using single cell lineage tracing information. And in particular, we were interested in those methods that give you shared state and lineage tree readout. There are a few different ones of those that can be categorized into prospective methods that actually engineer something into the cell, and retrospective methods that make use of naturally occurring mutations. You can use static barcodes or dynamic barcodes, usually using CRISPR-Cas editing systems. And on the retrospective side, you can either use naturally occurring mutations in nuclear DNA, or you can use them in mitochondrial DNA. And this project, we focused on dynamic barcoding technologies, such as the Linnaeus method, for which we also collaborated here with Bastian. And this is one of these methods where you engineer a barcode into cells together with an editing system. And then as time goes on, this barcode is stochastically engineered by your CRISPR-Cas9 system, such that over time, barcode complexity increases, and you can use these barcodes later on that you read out together with gene expression information to reconstruct lineage information across cells. So here's a cartoon of what that looks like in a simplified situation. So say we have just 2 cells. We start with 2 barcoded cells at time point t0 in 2 replicates. In the first replicate, we let the cell grow and divide a little bit. It accumulates 1 mutation. And in the end, we sequence the cells and we read out gene expression and the mutated barcodes. For the other replicate, we wait a little bit longer. So we accumulate more mutations. And in the end, we sequence again. Some cells we observe and other cells we will not observe. The readout we get from this is the mutated barcodes from both individuals, as well as gene expression information. Now, we can compare gene expression between the 2 time points as we did before in the Waddington OT approach. as implemented in Moscot now. But for barcode information, that doesn't work because these barcodes between the two different individuals cannot be related. They're randomly occurring mutations. So if two barcodes are similar within one individual, that has a meaning that says something about lineage relatedness. But if two barcodes are similar or not similar between two different individuals, that doesn't mean anything. Still, we wanted to be able to use this extra information to map across time points in vivo time series data sets. And for this, we developed the moscot-lineage method, which basically takes as an input the gene expression matrices, as well as barcode information, and potentially some marginals, which could represent something about uncertainty that you have about your cells or so. The output is a coupling, as we saw it before. It's essentially the same as in the simple case where you just have gene expression information linking cells from the earlier time points with cells from the later time point. We do it by providing the algorithm with two sources of information. So the first one lives in barcode space. So we have the two time points, and we are in a situation again where we cannot make one-to-one comparison between barcodes across the two different time points, but we can define cost functions, or we can define a cost metric between barcodes in the same individual. So we have one cost metric C1 here that lives in time point T1, and that we can use to compare barcodes there and to say something about lineage relatedness in that space. And we have another cost function C2 that compares barcodes at time point T2. In addition, we have gene expression information from these cells, because these methods give us a shared readout. And here we can make one-to-one comparisons. So we can ask how similar or dissimilar are cells in the shared gene expression space. And that naturally leads to a fused Gromov-Wasserstein problem again, where we have a Gromov term defined in barcode space, and we have a Wasserstein, a classic OT term defined in gene expression space, and we add entropic regularization. Once you have computed a coupling, you can actually feed this into CellRank. So there's an interface from Moscot to CellRank to infer state trajectories or to learn something about putative decision driver genes. So a quick detour. Actually, first I want to show how this works in Moscot. So again, this is extremely similar. Once you're familiar with the Moscot API, once you know it for one problem, it's really easy to do it for another problem. So again, you import this lineage mapping problem. You set it up by feeding it your anddata object. You prepare the method, and then you just hit solve. Now you pass, in addition to your entropic regularization, you know, pass the alpha parameter, giving the weight between the Wasserstein and the Gromov-Wasserstein terms. And Zoe has been involved in has been pushing the implementation here a lot. Okay, quick detour about how this works in CellRank, because originally CellRank was designed for RNA velocity data, but we recently extended it to be more applicable to different situations. So we also modularized CellRank into kernels that compute a cell-cell transition matrix, for example, between two different time points, and estimators, which can be used to basically do inference based on this cell-cell transition matrix, tell us something about initial states, terminal states, and fate probabilities. In CellRank 1, we had RNA velocity as a kernel, as well as just similarity and undirected kernel. And we had the GPCCA estimator that we used to compute these initial and terminal states and to say something about how likely macro states are to transition into one another. Now in CellRank 2, we have a pseudo-time, a cytotrace, and a time series kernel in addition to a new estimator. And the time series kernel, that is the interface to Moscot. So once you have computed a coupling between time points and Moscot, you can then feed this into the time series kernel in CellRank for further downstream analysis. And for the CellRank 2 project, I want to highlight Philip, who's co-leading this with me, and Mike, who's again really involved in the implementation. Okay, now I want to look more into the moscot-lineage method specifically, and I want to start by comparing it to currently the only other available approach that you can also apply to lineage-traced single-cell data in case it originates from these dynamic CRISPR-Cas barcoding systems where you cannot make one-to-one comparisons between barcodes across time points. So lineage OT is also based on optimal transport, but it's a different optimal transport variant. It's the classic optimal transport variant. So in this case, we assume that we have access to reconstructed lineage trees at both time points. We actually only use the later lineage tree. And And the later lineage tree is used to sort of shrink cells towards each other, which are related by lineage. And that is a trick to estimate the ancestor states of your observed cells at the later time points. That is the first step. And then in the second step, you link these inferred ancestor states at the second time point to your actual observed measurements at the previous time point. So it's essentially a pooling step connected with a classic optimal transport problem. So this does not make use of a Gromov-Wasserstein objective function as moscot-lineage does. To make comparisons here, we went to TEDSIM, which is a simulation tool for lineage-traced single-cell data. So you can input a state tree, and what it gives you is a simulated lineage tree, which we cut at 2 different levels to simulate 2 different time points. And as an output, you get simulated lineage barcodes that correspond to these lineage trees. You can tune the noise level that you want to have in these barcodes, and you get shared gene expression readout. We compared lineage OT against moscot-lineage in terms of this mean error. So the mean error basically is the difference in between, well, the mean error consists of 2 things. It consists of something called descendant error and ancestor error. So you can say, given some cells at the earlier time point, I want to make a prediction of where they're going to go. And I can compare this to the ground truth given by the simulated lineage tree from TEDSIM. And I can take the opposite perspective. I can also say, given cells at a later time point, I want to predict their ancestors, and I want to make a comparison to ground truth again. And here we saw that this mean error, so the mean error is then the average of these 2 errors and across different step sizes. So this is one of the simulation parameters in TEDSIM. We saw that moscot-lineage gives you much lower mean error. And we saw something very similar when we increased the stochastic silencing rate. So that's the noise level that essentially goes into these barcodes. And as expected, as you increase this noise level, the mean error goes up in both methods, but moscot-lineage gives you much lower mean error. Okay, so this was validation on simulated data. Next, we went into an application to real data. This is C.elegans early development, which is a very simple case because here the ground truth lineage tree is known and development is relatively deterministic. So there was no, in this experiment, there was no actual experimental lineage tracing done because the ground truth lineage tree is known. So cells were just assigned based on their transcriptome to ground truth, more or less known locations in this lineage tree. We compared moscot-lineage against a few different other methods across the different time points. These are time points past fertilization in minutes. And again, we used the mean error because again, we have access to the ground truth lineage tree in this simple animal to make comparisons. So we compare OT, classical optimal transport, which corresponds to alpha equals 0 in our moscot-lineage model. So that would only make use of transcriptomic information. Lineage OT as before. Gromov-Wasserstein, which would correspond to alpha equals 1 in our model. So that only makes use of lineage information and ignores transcriptomic information. And then moscot-lineage that sits somewhere in the middle and interpolates between using transcriptomic and lineage information. Across the time points, we saw that moscot-lineage either has the lowest mean error or the lowest mean, or second lowest mean error. So sometimes Gromov-Wasserstein had an even lower mean error. But in other situations, Gromov-Wasserstein has a really high mean error because it's really it's really affected if you have high noise level in your barcodes because it doesn't use transcriptomic information to regularize. But then we zoomed into one time point to the 210 to 270 minutes time point. That is a UMAP representation here. And we computed the difference in ancestor prediction error between lineage OT and moscot-lineage and saw that that difference was especially big for this small population here marked by this error. We then checked where this comes from. In the ground truth lineage tree, all of these cells actually originate from this one cell here, which is something that was recovered by moscot-lineage, but not by lineage OT. And we're not 100% sure yet about why that is the case, but there are many conceptual differences between moscot-lineage and lineage OT. One is, for example, that moscot-lineage makes use of gene expression of lineage information at both time points, whereas lineage OT only makes use of lineage information at the later time points, the later time point. We then zoomed out and computed a global coupling by chaining together the individual couplings across the time points, focused on this GLR population here. And used the computed coupling in CellRank to compute fate probabilities, which we then correlated with gene expression to infer these putative decision driver genes here that were also named in the original publication. So this is an example of how you can basically combine moscot-lineage with CellRank to reason about genes that could be driving a particular process. We have another application that we looked into for the moscot-lineage model. So this is a data set of zebrafish heart regeneration. So the question here is sort of how this process of heart regeneration plays out in the zebrafish. And it's an interesting model organism because in the zebrafish, the heart actually regenerates quite well, which is something that's not the case in humans. So in this experiment, the zebrafish heart is injured. You sequence at a control time point before the injury, and you have a few time points after injury where you sequence. In the original publication, Bastian and colleagues showed that fibroblasts play an important role in this regeneration process, which is why we wanted to zoom into these fibroblasts a little bit. So here's a UMAP of the various fibroblast populations in this data set. And we were interested especially in origin and importance of transient fibroblast populations. Because, for example, this collagen 11 population here only really exists at 3 days post-injury. So DPI stands for days post-injury. And it doesn't really exist at control time points. And it also really doesn't exist at later time points. And something similar holds for these collagen 12 fibroblasts. They don't really exist in the control time point, and they don't really exist 30 days post-injury. So there are transient populations that have probably an important role in the regeneration process. And we want to use moscot-lineage to learn something about their origin. Where do they come from and where do they go afterwards? In the original publication, they used a different method to reason about lineage origin, because in this data, they applied the Linnaeus method. So one of these dynamic CRISPR-Cas barcoding technologies. They reconstructed the lineage trees, and I then used a correlation-based analysis. So the idea being that if we correlate cell type proportions within each tree node across the entire tree, then on average, we would expect that if two cell types have really high correlation across these tree nodes, that means they have a shared lineage origin. So here's an example of what that looks like. That would be just doing this for a few fibroblast populations in one tree, within one tree. So each row corresponds to one cell type, and columns correspond to different tree nodes. And this heat map is colored according to the abundance of that cell type in that tree node. You can aggregate this in a scatter plot like this, where we just compare two different populations here, the collagen 12 fibroblasts in these epicardium cells. Each dot here would be a node in this lineage tree and would correspond to sort of how many cells did we see from cell type 1, how many cells did we see from cell type 2, and that gives you a correlation value between these two different cell types. They did this for all combinations of the different cell types they have at 7 days post-injury, and used hierarchical clustering of this heat map to learn something about lineage origin. So reassuringly, all the immune cells, they cluster together here, which makes sense because they actually have a shared lineage origin. And using this heat map and this clustering, you can also see that the collagen 12 fibroblasts, they are predicted to have a shared lineage origin with epicardium and with constituent fibroblasts, which was validated in the original publication with Krelog's lineage tracing. Now, with moscot-lineage, we wanted to go beyond this, but first we wanted to validate that it works in this setting. So we looked at the coupling matrix. That's a heat map of the coupling matrix you get when you apply moscot-lineage in this setting. And visually, one thing that's reassuring is that cell types seem to be linked to corresponding cell types across the two time points, which on average makes sense. For most cells, you would expect them to be linked to their corresponding cell type at the next time point. Of course, not for all cells. Some cells will change their cell type, but most cells you would expect to stay in the same cell type. So to formalize this notion a little bit, Bastian came up with the following test. Say we have control and we have 3 DPI. We have these 3 different populations at both time points. Let's say we focus on this blue population for now. then for each of the blue cells, we can look into the coupling that gives us basically, coupling tells us something about where this cell is linked to at control. So it provides a notion of this matching. And let's say we just take the 20 cells, for example, that are most closely matched to this blue cell here. Then this gives us a distribution over these 20 cells at control. So we get a distribution of the cell type of these 20 different cells at control. We can repeat this for all the blue cells and aggregate this. So this basically is something like, if I take the blue population at 3 dpi and I look at the predicted ancestors at the blue cells at control, what is their cell type distribution? And I can then compare this with the overall cell type distribution at control and use a statistical test to check whether any of these cell types is enriched compared to what I'd expect from just the distribution. So we use this test to predict the ancestor cell type of individual cell types at 3 dpi. And as I said, on average, we would expect that most cell types should be coupled to the corresponding cell type as an earlier time point. We did this, iterated over various decision thresholds to do this classification for various alpha values. And in general, we saw that this holds across the different cell types, across the two different time points very well. So moscot-lineage, in fact, does couple most cells from corresponding cell types. As you increase alpha, so alpha equals 0 corresponds to classic optimal transport. Alpha equals 1 is Gromov-Wasserstein here. So as you increase alpha, you use more lineage and less transcriptomic information. And as these clusters are defined in terms of transcriptomic information, of course, your accuracy to do this prediction goes down a little bit as expected. So this for us was validation that moscot-lineage works in this setting. Next, we tried to recover some of the predictions from the original paper. Using this test that I have just introduced, we were able to see that moscot-lineage predicts the ancestors of the collagen 12 cells at 3 dpi to be constituent fibroblasts. That's also something that they saw in the original publication based on the correlation test. Now with moscot-lineage, we could go a little bit further and also ask for the precursor cells of collagen 12 cells at 7 dpi. So we were asking what are the precursor cells at 3 dpi of these cells at 7 dpi. And moscot-lineage predicted these 3 different subsets of transient fibroblasts. That's something you could not have gotten by using the earlier, simpler correlation-based tests, because especially these collagen 11 cells, they're not really present any longer at 7 dpi, so you cannot perform this correlation. So that is an hypothesis that we can generate with moscot-lineage that you could not have formulated by using this simpler correlation-based analysis from before. Okay, here's a summary of the temporal applications. Moscot-temporal scales up the temporal mapping problem to larger cell numbers by having lower memory and time complexity. Moscot-lineage increases the accuracy of cell state transition determination, and moscot-lineage predicts activated fibroblast origin of the collagen 12 fibroblasts at 7 dpi in zebrafish regeneration. Okay. That brings me to the last part of this talk, which is mapping cells across space. So we consider 2 spatial applications as well. The first one I already introduced in the context of the NovoSpark method in the beginning, when I was motivating the fused Gromov-Wasserstein problem. So we have, as a reminder, we have the reference single-cell RNA-seq sample. We have our spatial target domain, where we have also measured expression of a few genes using any spatial transcriptomics method. If we solve this using an optimal transport-based method, if we solve this fused Gromov-Wasserstein problem, it gives us a coupling matrix relating cells in the reference single-cell RNA-seq sample with your spatial tissue. You can look at this from 2 different perspectives now downstream, either from a sample-centric perspective, in case you have cell-level covariates, like cell type information, for example. You can do a spatial deconvolution to assign these cell types to the different locations in the tissue. Or you can look at this actually from the feature-centric perspective, saying, I have a gene which I have measured in my reference here, but that is not contained in the spatial transcriptomics data. What is the likely expression of that gene in my spatial domain? So that's the feature-centric perspective. The different methods that span the spectrum, like Tangram and DEST-VI, that can do sort of both. NovoSpark, which can in principle do both because it covers this coupling matrix, but in the original publication focused on the feature-centric perspective, and Stereoscope and CellVI that focus more on the sample-centric perspective. Moscot sits in the middle, so it can also do both. It can give you the feature-centric perspective and also the sample-centric perspective. But in this talk, I'll just give a quick demonstration of what it can do in the feature-centric perspective. Again, here's a code snippet. This is, again, very easy to do in Moscot. You import the spatial mapping problem. You set it up with your two n-data objects corresponding to your reference and your spatial sample. And then you basically solve this problem. Okay, and Zoe has also been very much involved in this. So here's a benchmark that we did using now Moscot, and we benchmarked against the original NovoSpark, which solves the same fused Wasserstein problem, but using different cost matrices. So Marco talked a lot about the importance of these cost functions. So the cost function is defined a bit differently in NovoSpark than it is defined in Moscot. And also Moscot uses OGT under the hood, which enables the GPU acceleration and so on, as we saw before. So first we focused on a time benchmark. I should say that this is a seqfish data set. So the spatial data set is seqfish. The reference singles are RNA-seq. And in total, we have something like 30,000 cells here. We subsampled to various subsets and saw that moscot-lineage has much lower compute time than NovoSpark. If we go to this largest cell size here, corresponding to around 30,000 cells, this takes over half an hour if you run NovoSpark. It takes around 2 minutes if you run Moscot on this data set. Then we looked into the quality of the predictions a little bit. So what we did here is we basically took the original expression of these, I guess it was 351 genes that we had in the spatial sample, and we reconstructed them using Moscot, and then just checked Pearson correlation between the original values in the spatial domain and the predicted values. They increase as we increase the cell number. They're on average much higher in Moscot than they are in NovoSpark, which relates back to the definition of the cost function. And even though a correlation of 0.25 actually doesn't seem too high, qualitatively, they agree very well. And you should keep in mind that these correlations here are quite heavily influenced by random noise. So in a certain sense, you can say that there is a sense of denoising these values also. So even if the correlation isn't super high, qualitatively, we recover patterns that are quite similar between the original and the predicted expression, as you can see in this example here. Okay, and one of the powers actually of Moscot is that it has a very tight interface with SquidPy. So once you have recovered spatial gene expression, you can use SquidPy for both sample and feature-centric analysis, such as ligand receptor analysis, or looking for spatially informative genes, things like that. There's one other spatial application, which we call the spatial alignment problem. So the other one is the spatial mapping problem. In this case, we have two spatial samples. So now both of them have spatial information. Before we had one that only has molecular information and one that has molecular and spatial information. Now they both have spatial and molecular information. So here would be sample 1 that has a certain geometry. Here would be sample 2 that has a different geometry. And again, we are looking for a mapping between these two different samples. We can define a cost in terms of distance in Euclidean space, for example, in sample 1. We can define another cost in sample 2. And for those genes that are shared, in this case, I'm highlighting gene 1 and gene 2, I can make one-to-one comparisons between the two samples again. And this naturally leads to a fused Wasserstein problem again, with a Gromov-Wasserstein term and a Wasserstein term here, and entropic regularization. This problem comes in two different flavors, either the reference-based flavor, where you do direct comparisons between spatial samples. So it's like a spatial registration problem. Either you have different slices that come from adjacent regions in the same tissue and you want to compare them with one another, you can do that. Or you can align spatial samples with a pre-existing reference, so with a common coordinate framework. And it also comes in a de novo fashion, where given a collection of spatial samples, you want to reconstruct a common coordinate framework, so a template that you can use to jointly analyze all of these different samples that you have. We have a few different methods that sort of span the spectrum. There's some that can do both, like PASTE and GPSA. There's some that are more focused on the reference-based side of this. Moscot at the moment is also on the reference-based side, so it either needs a template to align to, or I can do sequential comparisons between different slides. But we're currently working on moving it here so that you can also do de novo template CCF construction from different samples. And this is also quite easy to do. In Moscot, it only comes down to importing spatial alignment problems, setting it up with your n-data object, and running it on your data. And Giovanni has been taking care of this implementation. Now we have a benchmark for that. We have a comparison with GPSA, which is a deep Gaussian process-based method that solves the same problem. So in this comparison, we adapted their simulation. So we have one sort of reference sample that consists of a certain geometry and random gene expression within it. We then create two data sets from that one data set by warping the spatial coordinates, adding random noise to gene expression, and subsampling these two data sets. And then we ask basically for the alignment of these two data sets that originated from this one original ground truth data set. So here's an example of what that looks like of the two samples. If you do that with Moscot, you get this left alignment here. If you do with GPSA, you get this right alignment here. So qualitatively, it looks like Moscot finds a very nice alignment here between these two spatial samples. Look into time. So far, we only run a CPU time benchmark. I suppose both methods can make use of GPU acceleration. So if you go to the GPU, probably both of them will perform better. But already on CPU, Moscot is quite fast for this problem, actually. And also in terms of mean squared error. So that relates to mean squared error between these two samples, because in an ideal world, you would be able to align them perfectly to each other because they originated from the same sample, right? We've just been adding random noise to them. Moscot gets a much lower mean squared error compared to GPSA. So here's a summary of the spatial applications. Moscot provides simple access to both the spatial mapping problem and the spatial alignment problem. It's faster than previous approaches, which enables application to much larger data sets as they're currently coming in. Modification of the cost terms leads to more accurate mappings in terms of the spatial mapping problem. And the SquidPy interface makes Moscot widely applicable to spatial data with various downstream analysis options. This brings me to the end of this talk, and I would like to thank the labs involved in this. Of course, Fabian's lab here, but also Moore's lab, where I did my lab exchange and where the Moscot project actually started. Dana's lab also, where I spent some time and where I did lots of work on CellRank. And then, of course, my funding partners. And I would like to thank you for your attention and for the opportunity for giving me the chance to present our work here. And I'm happy to take any questions.